thank to Birte for making my job easier. And now I'm going to tell you about the details of placing those sequences, which Birte skipped due to time constraints. Good afternoon, everyone. I'm going to present our study called Discovery and Genotyping of Novel Sequence Insertions in Many Sequenced Individuals. In this study, we are trying to answer the question, what is missing in the human reference genome? Our purpose is to discover novel sequence insertions in the whole genome sequence data because these sequences have important functional characterizations. They might be missing in the human reference genome because of two reasons. Either genome assembly errors occurred while generating the human reference assembly, or they are the deletions in the human reference. Discovering them will help to completely understand the human reference. Also, the allele frequency differences will help population scale analysis. Here is how an insertion sequence looks like. This is the reference sequence, and this is the donor genome with the insertion sequence. If we map the paired end reads of the donor genome to the reference genome, this is how they look like. We have 4 categories of reads. Concordant reads, where both ends of the reads map to the reference genome, and also the orientation of the reads and the insert size between the reads are as expected. Discordant reads, where again both ends of the reads map to the reference, but the orientation and the insert size are not as expected. Orphan reads, where none of the ends map to the reference, and one-end anchor reads, where only one end map and the other doesn't. For our purposes, we are going to use only orphans, one-end anchors, and concordant reads. Multiple tools are designed to solve this problem by analyzing next-generation sequencing reads. HiTSeq is considered as the first among such tools. It provides exact location range of breakpoints and partial insertion sequences, which is already a breakthrough in 2010, the time it is published. Later, Basil Ennis and Mind the Gap improved the results based on the similar concepts proposed in HiTSeq. They provide exact breakpoint locations and full insertion sequences. Recently, Poppins is developed to also support pooled sequencing data for population scale analysis and also genotyping all predictions. All of these tools start with read mappings, but they don't resolve multi-mapping issue well enough. In fact, the last three tools based on the results of underlying mappers, such as BWA, which in many cases report a subset of locations of multi-mapping loci. This decreases the performances on detecting events in repeat or duplication-rich regions. Now I'm going to talk about our method. We developed Pamir for novel insertion discovery in multiple samples. We start with read mappings, and we apply preprocessing operations to prepare the input for cluster formation. We generate the clusters with one-end anchors and orphan contigs. We assemble the clusters and map the resulting contig to the respective reference region to discover the insertion breakpoints and contents. We apply post-processing operations to filter false positives. Finally, we genotype the resulting call set. Here is the preprocessing and cluster formation in detail. We start with extracting one-end anchors and orphan reads from the best alignment output. We multi-map one-end anchors not to miss any information because of the repeats. We assemble orphan reads. In Pamir, we support both De Bruijn graph and overlap layout consensus graph-based assemblers. Next, we find an association between one-end anchors and orphan contigs through mapping. Then, according to their mates locations, we cluster the one-end anchors with the orphan contigs. The criteria for clustering is the rightmost mapping one-end anchor mate's location shouldn't exceed twice insert size distance from the leftmost mapping one-end anchor mate's location. After generating our clusters, we assemble them into potential novel insertion candidates with a strand-specific, strand-aware overlap layout consensus graph-based assembler, in which the reads are the nodes and the overlaps between the reads are the weights of the edges. It is a directed graph. The directed graph might have cycles, and we are searching for the maximum weighted path problem in the directed graph, but we remove the cycles when we observe, so it's turned into a maximum weighted path problem in directed acyclic graph. We apply a dynamic programming solution with topological ordering. By maximizing the total sum of the overlaps between the reads, we obtain a potential novel insertion candidate. After obtaining our potential insertion candidates, we map them to the respective reference regions. We extract the respective reference region according to the start and end locations of the cluster. We use a modified Smith-Waterman alignment algorithm which makes global to local alignment. It globally maps the insertion candidate locally to the reference. Here, our green candidate, which is a successful insertion, is mapped to the respective reference region. Its left and right flanks map reasonably long enough to the reference, and the middle part is the insertion itself, and the end of the left flank's alignment is the insertion breakpoint location. After obtaining our insertion candidates, we apply post-processing operations. After obtaining our insertion calls, we apply post-processing operations. First one is filtering. In filtering, we map all unmapped reads, one-end anchors plus orphan reads, to a template insertion sequence, which we generate by attaching the upstream of the breakpoint on the reference, the insertion itself, and the downstream of the breakpoint. We expect to see at least one pair of reads mapping to this template sequence such that the first mate is mapping to the reference and second mate is mapping to the insertion. We expect this for both of the insertion breakpoint locations. And if these conditions are not satisfied, we eliminate the insertion. But filtered results have another issue. In our experience, many of the false positives are due to mapping artifacts caused by multi-mapping reads. We resolve this problem based on the principle of maximum parsimonious approach, in which minimal number of insertions will represent all of the reads, which is an instance of set cover approach. Consider this example where 8 reads are mapped to 4 insertions. The red arrow represents a unique map, and the black arrow represents a multi-map. Consider the insertions as the sets and the reads as the universe. In set cover approach, the reads that map to multiple novel insertion sequences are assigned to the insertion which has the highest unique read support. As a result of maximum parsimonious approach, minimum number of insertions will represent all of the reads. So here in this example, insertion 2 is eliminated because it was supported by only multi-mapping reads. After obtaining our filtered calls, finally we genotype them. The genotyping strategy we used in Pamir is to compare the mapping results on the reference sequence and the hypothetical insertion sequence. We calculate the genotypes according to the following formula. It is a ratio between the number of reads crossing the breakpoints of the insertion and the number of reads crossing the breakpoint of the reference. According to this ratio and gamma, according to the relation between this ratio and gamma, the insertion is called either no insertion, homozygous variant, or heterozygous variant. Here in this example, the number of reads crossing the breakpoint of the reference is 2. The number of reads crossing the breakpoints of the insertion are 9 and 7, which makes 8, and the ratio between these two numbers is 0.6, which is greater than gamma, this insertion is called as homozygous variant. To recap, we start with extracting one-end anchors and orphan reads from the best alignment output. We multi-map one-end anchors. We assemble orphan reads into orphan contigs. We find an association between one-end anchors and orphan contigs. We generate the clusters. We assemble the clusters and map the resulting insertion candidates to the reference to obtain the insertion breakpoint location and insertion content. We apply post-processing operations, and finally, we genotype our calls. In fact, our design is so flexible such that with the following modification, Pamir can be extended to discover novel sequence insertions in multiple individuals from the same population. We start with, after getting the best alignment outputs for each sample, we extract the one-end anchors and orphan reads from each sample and combine them in two groups of one-end anchors and orphans. We follow the same procedure until genotyping, and at the end, we genotype each sample individually. Now I'm going to talk about our results. First, we designed the simulation inserted human reference sequence as follows. We inserted into human reference GRCH37, chromosome 21, 350 insertions, whose lengths differ from 5 to 10,000. We got the insertions from Methylobacterium, which are guaranteed to be missing in the human reference. As a result, these sequences will generate one-end anchors and orphan reads, and therefore are detectable by any computational tool. Then we simulated 6 high-coverage single genomes, 3 error-free and 3 noisy, with these sequencing models and these read lengths with ART simulator. We also generated 5 10x coverage, low-coverage genomes with Illumina HiTSeq 2500 100 base pair model. For these low-coverage genomes, we kept all 350 insertions in the first sample and deleted 70 randomly selected mutually exclusive insertions from the other 4. Here are the results of Pamir, Poppins, Mindigap, and Basil Annis on the single high-coverage genomes. Best results are shown in bold. We can say that Pamir outperforms the existing tools in all of the cases, in both precision and recall, except for the first 4 data sets, where Mindigap has equal precision with Pamir. We compared the results of low-coverage samples with Poppins, which is the only available tool for multi-sample support. In the first table, we show the precision and recall rates on the individual runs. Best results are shown in bold. Pamir is doing better than Poppins. If we do pooled calling with all of the samples, there is precision and recall are shown on the second table. Pamir is still doing better, but the recall rate is significantly higher than the individual runs. When we check genotyping accuracies of these individuals of the pooled run, the genotyping accuracies are pretty high. Both Pamir and Poppins are doing equally well. Only 5 calls of 2 breakpoints are incorrectly genotyped by Pamir, both of which fall in repeat regions. And all of Poppins' calls were correctly genotyped. As real data experiment, we use CHM1, which is a haploid genome and is suitable for structural variation detection. It is a special data set that has both short Illumina reads and long PacBio reads. PacBio calls are made by SMRT-SV tool by Chassin and Huddleston et al. And according to the short Illumina reads, which Pamir is designed for, majority of the calls are short and or homozygous, although all of them should have been homozygous. We also ran Pamir on 10 low-coverage genomes of 1,000 genome samples, but I'm not going to present the results here. In summary, we developed a new tool, Pamir, for novel insertion discovery. It effectively uses one-end anchors and orphan reads. It provides the exact insertion breakpoint location and insertion content. It also provides genotyping information and supports multiple samples. And according to our tests on simulation data, it outperforms the existing tools. And this concludes my talk. I would like to thank my collaborators and advisors. And travel funding to ISCB 2017 was generously supported by HiTSeq, ISCB, and EMBO. Thank you for your interest.
Questions?
Thank you. I have a quite general question about this, this research. So, do you have any kind of idea of which kind of sequences these novel sequences are in terms of whether they have coding potential, whether they are located at regulatory elements? I mean, which kind of functionality they have?
Oh, we have no idea what kind of functionality they have because no one analyzed the functions of these insertions, but they can be anywhere, like in non-coding regions or also coding regions. So maybe the ones in the coding regions will be very important for all cases. But we haven't analyzed any of these sequences, just found them.
Which is the fraction of the genome of one individual that will be comprised of these new sequences? It's kind of 0.0001% or? Oh,
I have no idea about the fraction. So.
Okay. Thank you.
Because we didn't check which regions do these sequences fall in. Yeah, we didn't annotate them.
Okay, thanks.
Let's move on and thank the speaker again.